Basics of Medical Biochemistry. First edition, 2022. Chapter 3. Chemistry of Carbohydrates, Part 2 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tao, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. 3.9 Some important disaccharides. Disaccharides are those sugars that yield two same or different molecules of monosaccharide on hydrolysis. Their general formula is CN, H2O, There are two basic types of disaccharides: reducing disaccharides, in which the monosaccharide components are bounded by hydroxyl groups, and non-reducing disaccharides, in which the components bound through their anomeric centers. The two monosaccharides are bonded via a dehydration reaction, also called a condensation reaction or dehydration synthesis, that leads to the loss of a molecule of water and the formation of a glycosidic bond. Table 3.4 Common disaccharides. Disaccharide. Unit 1. Unit 2. Bond. Type. Sucrose, table sugar, cane sugar, saccharose, or beet sugar. Glucose. Fructose. Alpha, 1 rightward arrow 2. Non-reducing. Lactose, milk sugar. Galactose. Glucose. Beta, 1 rightward arrow 4. Reducing. Maltose. Glucose. Glucose. Alpha, 1 rightward arrow 4. Reducing. Trehalose. Glucose. Glucose. Alpha, 1 rightward arrow 1. Non-reducing. Cellobiose. Glucose. Glucose. Beta, 1 rightward arrow 4. Reducing. 3.9.1 Maltose. Maltose or malt sugar, the crystalline disaccharide, is an intermediary in acid hydrolysis of starch and can be obtained by enzyme hydrolysis of starch. Sources. The main dietary sources of maltose are rice, potatoes, cereals, and malt. Maltose is produced from starch by hydrolysis in the presence of diastase, an enzyme present in malt. The production of maltose from germinating cereals, such as barley, is an important part of the brewing process. When barley is malted, it is brought into a condition in which the concentration of maltose-producing amylases has been maximized. Mashing is the process by which these amylases convert the cereal's starches into maltose. Metabolism of maltose by yeast during fermentation leads to the production of ethanol and carbon dioxide. Structure. It has the same empirical formula, C12H22O11, as sucrose and lactose, but differs from both in structure. It is formed by two units of glucose joined with an alpha, 1 rightward arrow 4, linkage. The glycosidic linkage is found between the carbon number 1 of one alpha-D glucose with the carbon number 4 of another glucose. That's why the linkage is termed as alpha, 1 rightward arrow 4, type. Chemically it is O-alpha-D glucopyranosyl 1,4, alpha-D glucopyranose. Figure 3.21 Structure of maltose showing alpha, 1 rightward arrow 4, glycosidic linkage. Characteristic features of maltose. Maltose is a disaccharide of two glucose units, present in malt and malt products. It is produced within the body by the action of salivary amylase, in the oral cavity, and pancreatic amylase, in the small intestine, upon starch. Maltose is further hydrolyzed to two glucose units during digestion by the enzyme maltase present in the small intestine. Maltose is a reducing sugar as one potentially active aldehyde is still found on one glucose component. The maltose may be alpha or beta depending upon the position of the O group on the anomeric carbon atom of second glucose. It produces a bunch of flowers like crystals of ozazone when treated with phenylhydrazine. Isomaltose is the type of maltose in which glycosidic linkage is alpha, 1 rightward arrow 6, type IE. An oxygen bridge is found between carbon number 1 of one glucose with the carbon number 6 of another glucose. Isomaltose is found in glycogen, amylopectin and certain bacterial dextrin, at branch point. The molecular mass of maltose is 342.30 g/mol, density is 1.54 g/cm3 and melting point is 102 to 103 degrees Celsius. Isomaltose is hydrolyzed to 2 units of glucose in the intestinal tract by the enzyme oligo 1,6 glucosidase, isomaltase. Clinical aspects: plain maltose has a sweet taste, about half as sweet as glucose and about 1/6 as sweet as fructose. The most common way to consume it is to put a layer of maltose between 2 pieces of biscuits, usually crackers. Food rich in maltose is easily digested. Pompe's disease: it is a type of glycogen storage disease. It is also an autosomal recessive disease due to a deficiency of enzyme acid maltase, 1,4 glucosidase, present in the lysosome. It is characterized by excessive glycogen concentration accumulated in abnormal vacuoles in the cytoplasm. The main symptoms are muscle hypotonia and severe cardiomegaly, and death occurs early. 3.9.2 Lactose. Lactose is a disaccharide known as milk sugar formed by 1 unit of glucose and 1 unit of galactose joined with alpha, 1 rightward arrow 4, linkage. The chemical name of lactose is beta-D-galactopyranosyl-, 1 rightward arrow 4, D-glucose. Sources: lactose is abundantly found in milk and dairy products. It is produced by the mammary glands of humans and most mammals. It is synthesized within the mammary gland and occurs as a 5% component of milk. The hydrolysis of lactose yields 1 molecule each of glucose and galactose. The enzyme responsible for this process is called lactase, present in the intestine. Structure: in the molecule of lactose, the galactose and glucose moieties are linked together utilizing beta, 1 rightward arrow 4, glycosidic linkage. The glycosidic linkage is between carbon number 1 of beta-D-galactose and carbon number 4 of alpha-D-glucose. Figure 3.22 Structure of lactose showing, 1 rightward arrow 4, glycosidic linkage. Characteristic features of lactose. It is a reducing sugar, that forms ozazone crystals of the cotton ball like on reaction with phenylhydrazine. It is not soluble in water, dextrorotatory in nature and least sweet sugar being 16% of sucrose. It is hydrolyzed by lactase in the intestinal tract into glucose and galactose units. Lactose plays a major role in the synthesis of milk in lactating mammary glands. The process of synthesis of lactose is responsible for drawing water into milk as it is being formed in mammary epithelial cells. This water is sufficient to meet the infant's needs for sweating and respiratory water loss. Lactose is a major readily digestible source of glucose which provides energy to the neonate and provides galactose to the developing brain for synthesizing glycolipids. It enhances the absorption of calcium, magnesium, and zinc from milk. Lactose is a substrate for many bacteria, Lactobacillus acidophilus and Lactobacillus bulgaricus, which ferment lactose into lactic acid. These lactobacilli colonize the gastrointestinal tract of the infant as a protective factor against other bacterial toxins, occupying a limited number of binding sites along the intestinal wall. Bifides factor is identified in humans. Lactose is responsible in part for the milk volume, and lactating mothers need an adequate source of lactose in their diet. Human milk differs from cow's milk in containing L-fucosyl lactose, in addition to regular lactose, in which deoxy sugar L-fucose is linked to galactose at carbon number 2. Clinical aspect: lactose intolerance. It is the inability to digest a significant amount of lactose due to the deficiency of the enzyme lactase. As a result, lactose is not broken down into simple sugar to be absorbed into the blood. Following are the causes of lactose intolerance. Primary lactase deficiency. It is the most common form of lactose intolerance. It is genetically inherited and develops within 5 to 20 years of age when lactase production decreases due to decreased milk in the diet. 2. Secondary lactase deficiency. It occurs as a result of any illness, injury, medication, or surgery. Conditions that can cause it are celiac disease, Crohn's disease, gastroenteritis, and ulcerative colitis. Treatment and medications that can be considered to manage this disease are chemotherapy and long-term use of antibiotics. 3. Congenital lactase deficiency. It is a rare genetic disease in which a minute quantity of lactase is produced. 4. Familial lactase deficiency. It is similar to congenital lactase deficiency. Here the body can produce a substantial amount of lactase but it is unable to break down lactose properly. Symptoms: nausea, flatulence, diarrhea, bloating and gurgling in the stomach associated with pain and abdominal cramps. Diagnosis: it is based on the following tests. I. Lactose intolerance test. The test begins with the individual in a fasting condition. The person takes a lactose-rich drink. Several blood samples are obtained over 2 hours. Mechanism: normally when lactose reaches the small intestine, the enzyme lactase breaks it down into glucose and galactose. The liver then converts galactose to glucose which enters the bloodstream raising the blood glucose level. If lactose is incompletely broken down, blood glucose level does not raise indicating lactose intolerance. 2. Hydrogen breath test. The patient drinks a lactose-loaded drink and the breath is analyzed at regular intervals. Raised levels of hydrogen in the breath indicate improper digestion of lactose. Mechanism: the undigested lactose in the colon is fermented by bacteria resulting in the production of various gases including hydrogen. The hydrogen is absorbed from the intestines, carried through the bloodstream to the lungs and finally exhaled. In case of lactose intolerance, hydrogen is accumulated in the blood. 3. Stool acidity test. It is carried out in infants and young children. It measures the amount of lactic acid in the stool. Mechanism: undigested lactose fermented by bacteria in the colon creates lactic acid and other short-chain fatty acids that can be detected in the stool sample. 4. Small bowel biopsy. A piece of tissue is taken from the small intestine with the help of an endoscope. The amount of lactose is then measured. This test is rarely carried out. Treatment: by eliminating lactose from the diet and abstinence from all dairy products such as ice creams, butter, cheese, yogurt, chocolates, peanut butter and bakery items. Complications: main complications include osteopenia, osteoporosis, malnutrition, and weight loss. Lactasuria: the presence of milk in the urine is called lactasuria. It is frequent during pregnancy and lactation. This condition is identified by characteristic ozazone crystals. Lactulose, a synthetic disaccharide, beta-1,4-galactosidofructose which is neither hydrolyzed nor absorbed in the human. It is metabolized by the intestinal bacteria with the production of organic acids. It is used in the treatment of encephalopathy due to advanced cirrhosis of the liver. The unabsorbed sugar produces diarrhea and the acid pH help to contain ammonia in the feces. Clinically it is used as an effective laxative to relieve constipation. Galactometer. Lactometer: it is a device used to check the amount of water present in the milk, by measuring specific gravity. Galactorrhea, excessive flow of milk. Galactotherapy, treatment of disease using an exclusive or nearly exclusive milk diet. 3.9.3 Sucrose. Sucrose, common name is table sugar or cane sugar, also called saccharose, is a non-reducing disaccharide. On hydrolysis, it gives one molecule each of fructose and glucose. Sources: sucrose is obtained from cane sugar, sugar beet, and sugar maple. It occurs in the juices of most vegetables and fruits, example pineapples, sugar cane, sugar beet, and carrots. Structure: it is formed by one molecule of D-glucose and one molecule of D-fructose joined with alpha, 1 rightward arrow 2, glycosidic linkage. Figure 3.23 Structure of sucrose showing alpha, 1 rightward arrow 2, glycosidic linkage. Characteristic features of sucrose. It is by far the most abundant and sweetest sugar. It is non-reducing sugar as there is no free aldehyde or ketone group, as the glycosidic linkage is found between carbon number 1 of glucose and carbon number 2 of fructose. During the hydrolysis of sucrose, there occurs a change of optical rotation from plus Ve to Ve as D-fructose is more level rotatory than D-glucose. Hydrolysis of sucrose by dilute acid or by sucrase, invertase, produces one molecule each of glucose and fructose. Sucrase. Downward arrow. Sucrose plus H2O glucose plus fructose. Plus 66.50 plus 52.50 92O. Because of the inversion of the sign, the process of hydrolysis of sucrose is called inversion and the mixture of glucose plus fructose, so obtained is called invert sugar. The sucrose is first split into D-glucopyranose, plus, and D-fructofuranose, but the latter is changed into D-fructopyranose, which is a very strong level rotatory sugar. Sucrose is the most easily hydrolyzed sugar. It is suggested that the rate of acid hydrolysis of lactose is taken as 1, then the rate for maltose is 1.27 and for sucrose is 1240. Its molecular mass is 342 g/mol, density is 1.587 g/cm and melting point is a 186 degrees C. Sucrose is readily fermented by yeast. It does not form ozone crystals unless hydrolyzed. Does not show mutarotation. Sucrose and other disaccharides can't be utilized when given intravenously. Clinical aspect: sucrose intolerance. The body cannot metabolize sucrose due to the deficiency of enzyme sucrase. Important clinical features are abdominal pain, nausea, vomiting, diarrhea, flatulence and bloating. It is managed by eliminating sucrose from the diet. Dental caries. Overconsumption of sucrose has been linked with some adverse health effects. The most common is dental caries or tooth decay, in which oral bacteria convert sugars, including sucrose, from food into acids that attack tooth enamel through the process of demineralization. Intravenous sucrose, however, cannot be utilized by the body and will be excreted in the urine except in patients with cerebral edema, where it changes the osmotic condition of the blood and causes a flow of water from the tissues into the blood, relieving the symptoms. When a large amount of food that contains a high percentage of sucrose is consumed, beneficial dietary effects are displaced from the diet, which can contribute to an increased risk for chronic disease. It has been suggested that sucrose-rich dishes and drinks may be linked to the development of obesity and insulin resistance. The rapidity with which sucrose raises blood glucose can cause problems for people suffering from defects in glucose metabolism, such as persons with hyperglycemia and diabetes mellitus. 3.10 Some important polysaccharides. Polysaccharides molecules have many actual or potential sugar groups. Poly equals many, saccharide equals sugar. These compounds are made up of a large number of simple sugars linked by a glycosidic bond. Polysaccharides yield a large number of monosaccharide units upon hydrolysis. 3.10.1 Homopolysaccharides, polymers of the same monosaccharides. These polysaccharides are made up of the same kind of monosaccharide units. These compounds yield the same type of monosaccharide units upon hydrolysis. 3.10.1.1 Starch. Starch is a homopolysaccharide commonly found in plants. It is non-reducing in nature and insoluble in water. It forms colloidal solutions upon dissolving in water. It is a high molecular weight compound. Sources: potato, legumes, cereal, wheat, and carrots along with the seeds, leaves and stems of most vegetables are rich in starch. Structure: it consists of two types of polysaccharide units, amylose and amylopectin, which in turn are made up by the polymerization of a large number of alpha-D-glucose molecules. It is a polyglucose having, one rightward arrow 4, glycosidic bond among glucosyl residues, also called glucosan or glucan. In the amylose, carbon number 1 of the first glucose is attached to carbon number 4 of the second through an oxygen bridge with the elimination of a water molecule. It is a hydrated micelle in the form of a twisted helical coil. Amylose has 50 to 5,000 glucose units and it constitutes 15 to 20% mass of starch. It gives a blue color with iodine. Figure 3.24 Structure of a segment of amylose molecule. Amylopectin is another component of starch and it is more abundant than amylose. It forms 80 to 85% of starch. It is made up of two types of linkages. In a linear chain, the linkages are the same as those in amylose, one rightward arrow 4 linkages, and account for 96 to 97%, but at the branch point, there is one rightward arrow 6 bond which results in the branching of the molecule. In one rightward arrow 6 linkage, carbon number 1 of the terminal glucose molecule of the straight chain is linked to carbon number 6 of non-terminal glucose residue present in the main chain. Each straight chain contains about 24 to 30 glucose units. The amylopectin molecule is much bigger and more complicated than amylose, as it may have up to 1 million glucose units. It forms a colloidal solution which gives a purple or bluish-violet color with iodine. Amylopectin can be represented as shown in Figure 3.25. Figure 3.25 Structure of a segment of amylopectin molecule, showing many alpha, 1 rightward arrow 4, linkages and 1 alpha, 1 rightward arrow 6, glycosidic linkage at the branching site. Characteristic features of starch. It is the main form of carbohydrates taken by the human diet. It is the storage carbohydrate in plants. As free aldehyde groups are not available in starch, so it does not act as a reducing agent and is unable to form ozone crystals. Starch is a dietary polysaccharide and on hydrolysis produces glucose. Glucose is the major metabolic fuel of the body. The action of acids hydrolyzes starch to glucose by breaking both 1 rightward arrow 4 and 1 rightward arrow 6 glycosidic linkages. Starch passes through the following intermediates which give particular color to reactions with iodine. Insoluble starch, in cold water, blue. Soluble starch, blue. Amylodextrin, blue-purple. Erythrodextrin, red. Acrudextrin, no color. Maltose, no color. Glucose, no color. Significance of starch. Starch is the main dietary carbohydrate that provides glucose to the body. Digestion of starch begins from the mouth. The saliva contains the enzyme salivary amylase, also called ptyalin, which hydrolyzes starch by breaking the glycosidic linkage present between glucosyl residues to produce dextrin, maltotriose, and maltose. This process is continued in the small intestine where pancreatic amylase is present in sufficient quantity. Various disaccharidases of the intestinal juice hydrolyze the disaccharide units into simple monomeric units, and finally, glucose is produced which is absorbed in the bloodstream. Table 3.5 Difference between amylose and amylopectin. Amylose. Amylopectin. Unbranched structure, having glucose arranged in linear chains. 1 rightward arrow 4, linkages are present. Water soluble. Contributes 15 to 20% mass of starch. Low molecular weight. Highly branched. Both 1 rightward arrow 4 and 1 rightward arrow 6 linkages are present. Water insoluble. Contributes 80 to 85% mass of starch. High molecular weight. 3.10.1.2 Glycogen. It is another polymer of glucose regarded as a storage polysaccharide of the animal cell. It is therefore also called animal starch. Sources: glycogen is chiefly found in the liver and skeletal muscles. It is also present in other cells of the body but to a lesser extent. These include kidneys, glial cells in the brain, red blood cells and white blood cells. Glycogen is mainly distributed in the cytoplasm of the cell as discrete granules, and a small amount is also present in lysosomes. Structure: glycogen is the analog of amylopectin of starch. It is a highly branched polymer of alpha-D-glucose having both alpha, 1 rightward arrow 4, glycosidic bond in the linear chain and alpha, 1 rightward arrow 6, glycosidic linkage at the branch point. In the molecule of glycogen, 92% of the linkages are alpha-1,4 types and 8% are of alpha, 1 rightward arrow 6, type. Glycogen is more compact and highly branched than amylopectin. Branches occur after every 8 to 12 residues. Repeated branches give it a tree-like structure. There are about 55,000 glucose residues in 1 unit of glycogen. As each branch ends with non-reducing sugar, this polymer has many non-reducing ends but only one reducing end. Figure 3.26 Structure of glycogen, glucose linked by alpha, 1 rightward arrow 4, glycosidic bond in the chain, glucose linked by alpha, 1 rightward arrow 6, glycosidic bond at the branch point, reducing end, non-reducing end. Characteristic features of glycogen. Glycogen is abundant in the liver, 100 grams, comprising 8 to 10% of the fresh mass of well-fed liver in adults. About 400 grams of glycogen is present in skeletal muscles which make up to 1 to 2% of their mass. Muscle glycogen is regarded as a fuel reserve for the synthesis of ATP during muscle contraction. Liver glycogen is responsible to maintain blood glucose levels, particularly during the early stages of fasting. The molecular mass of a single molecule of glycogen is up to 108 daltons. It is also found in non-green plants, fungi and yeasts, having no chlorophyll. It is non-reducing, readily soluble in water and gives a red color with iodine. Clinical aspects. Glycogen storage diseases are a group of genetic disease that results from a defect in an enzyme required for glycogenesis, glycogen synthesis, or glycogenolysis, degradation of glycogen. The balance in the rate of glycogenesis and glycogenolysis is responsible for maintaining blood glucose levels. As a meal containing carbohydrates is eaten and digested, insulin stimulates the action of glycogen synthase, due to which glucose molecules are added to the chains of glycogen until the amount of insulin and glucose become balanced. About 4 hours after a meal, glycogen phosphorylase starts to break down glycogen into glucose. 3.10.1.3 Cellulose. Cellulose is the most abundant organic matter in the world, chiefly found in the plant kingdom and comprising more than half of all the organic carbon. Sources: it is present mainly in the cell wall of plants. It is the main constituent of plant stems, stalks, trunks, etc. All the wood and cotton are almost pure cellulose. Structure: it is, like amylose, an unbranched polymer of beta-D-glucopyranose consisting of 10,000 to 15,000 residues joined to each other through beta, 1 rightward arrow 4, glycosidic linkages. It is made up of repeating units of the disaccharide called cellobiose. Cellobiose has the same structure as maltose, but despite alpha, 1 rightward arrow 4, glycosidic linkage, cellobiose has beta, 1 rightward arrow 4, type of glycosidic bond. The beta configuration allows cellulose to have very long straight and linear chains. The parallel chains of cellulose give rise to fibrils that have high tensile strength. Its average molecular weight is around 570,000. Characteristic features of cellulose. It provides fiber and bulk to the food. This roughage serves to satisfy the appetite, and in addition, it stimulates the intestinal peristalsis. It has no nutritional value as it is not digested in the human gastrointestinal tract because a man lacks the enzyme cellulase. Herbivorous animals also do not secrete cellulase in their digestive juices, but they can utilize cellulose by symbiotic intestinal microorganisms. It can be synthesized in the laboratory. Substitution of O group of carbon number 2 by an acetylated amino group gives rise to chitin, which is present in the exoskeleton of insects and crustaceans. Clinical aspect: although cellulose remains undigested in the human body and unable to act as a source of energy, but still it is important to have a good amount of fiber in diet because it prevents constipation, helps in the digestion of other food materials, stimulate and regulate intestinal peristalsis, regulates water absorption in the gastrointestinal tract, reduces the chances of occurrence of cancer in the colon. 3.10.1.4 Hemi Cellulose. It is a family of high molecular weight carbohydrates having a structural resemblance to cellulose, but it is more soluble and more easily decomposed. It is the chief constituent of the plant cell wall. It can be extracted with dilute alkalis and is precipitated by dilute acids. Various sugars including aldopentoses, uronic acids, and xylans are produced from the hydrolysis of Hemi Cellulose. Wood, straw, and bran have a significant amount of xylan, a polymer of xylose. 3.10.1.5 Dextrin. Dextrins are produced during the partial hydrolysis of starch by enzyme amylase or by acid or heat. They give different colors with iodine as insoluble starch, in cold water, blue. Soluble starch, blue. Amylodextrin, blue-purple. Erythrodextrin, red. Acrodextrin, no color. Dextrins are composed of a very complex mixture of molecules of different sizes and structures. They occur in the leaves of all green plants as intermediates either in the synthesis of starch from glucose or in the degradation of starch. They generally have a sweet taste. Alpha-dextrins have on average 8 alpha-D-glucose residues showing mostly alpha, 1 rightward arrow 4, glycosidic linkages in addition to an alpha, 1 rightward arrow 6, linkage at each of the branching sites. Limit dextrin is a well-defined dextrin produced after the action of amylase on dextrin. They form sticky solutions in water and are frequently used as adhesive, example on postage stamps and envelopes. 3.10.1.6 Dextrans. These are alpha-D-glucopyranose polymers, having mostly alpha, 1 rightward arrow 6, linkages but also have, 1 rightward arrow 2, 1 rightward arrow 3, and 1 rightward arrow 4, linkages between adjacent glucose units. Dextrans occur in nature forming an adhesive coat around certain bacteria. Their solutions are highly viscous. These polysaccharides are synthesized by Leuconostoc mesenteroides and some other microorganisms in culture media having sucrose. Clinical aspects. Dextrans, having a molecular weight from 25,000 to 75,000, are used as a plasma substitute or expander in the treatment of hypovolemic shock. As it remains in the blood for many hours and increases the blood volume because of its high viscosity, low osmotic pressure and slow disintegration. However, it can interfere with blood grouping and cross-matching. That's why blood sample for the blood grouping is collected before the administration of dextrin. 3.10.1.7 Inulin. It is a homopolymer of D-fructose found in tubers and roots of dahlias and dandelions. It is tasteless, water-soluble, and levorotatory in nature. It gives no color with iodine. Its molecular weight is about 5,000. Inulin has no nutritional value as a human has no inulinase, present only in plants. Clinical aspect. Clinically it is used in the determination of glomerular filtration rate, GFR, and estimation of the volume of body water. 3.10.2 Heteropolysaccharides, polymer of different monosaccharides and other substances. The heteropolysaccharides are made up of different kinds of monosaccharide units and they yield the dissimilar type of sugars as well as other non-carbohydrate residues upon hydrolysis. There are three main groups of heteropolysaccharides, i.e., glycosaminoglycans, GAGs, or mucopolysaccharides, glycoconjugates, and mucilage. 3.10.2.1 Glycosaminoglycans, GAGs, or mucopolysaccharides. Glycosaminoglycan molecule is an unbranched polysaccharide made up of repeating units of disaccharides. These are primarily located on the surface of cells or in the extracellular matrix. GAGs are highly negatively charged molecules, with high viscosity to the solution, so they are ideal for a lubricating fluid in the joints. They are generally composed of an amino sugar as hexosamine, N-acetylglucosamine or N-acetylgalactosamine, and uronic acid, usually D-glucuronic acid or L-uronic acid. They also possess sulfate groups in many cases. Their specific patterns of organization render them easy recognition by a variety of ligands. Figure 3.27 Pattern of structural organization of repeating disaccharide unit, acidic sugar plus amino sugar, in glycosaminoglycan. Glycosaminoglycans have the special property to bind a large amount of water to produce a gel-like matrix that provides the basis of the ground substance of the body. They stabilize and support cellular and fibrous components of tissue and involve in maintaining the water and salt balance of the body. The cell-cell interaction is also mediated by glycosaminoglycans. Following are the important types of glycosaminoglycans. I-hyaluronic acid or hyaluronate. Hyaluronic acid is a very large, high molecular mass, more than 1 million, chain-like molecule. It was first isolated from the vitreous humor of the eye. It is now found in many other tissues including cartilage, loose connective tissues, skin, umbilical cord, synovial fluid, hemolytic streptococci, etc. It acts as an excellent lubricator and shock absorber. It has a significant role in morphogenesis and cell migration during embryogenesis. Structurally, it is composed of repeating units of D-glucuronic acid and N-acetylglucosamine. There are up to 50,000 repeats of this pair. Hyaluronan contains no sulfate and is attached to proteins in proteoglycan by salt-like bonding. Figure 3.28 Structure of hyaluronic acid. Hyaluronidase is an enzyme that can break hyaluronic acid and by reducing its viscosity helps in the diffusion of materials into the tissue space. It is found in certain bacteria, the sting of bees and the venom of the snake. Bacterial hyaluronidase increases its penetrating power by destroying the intercellular hyaluronic acid barrier. Hence, sometimes it is named as spreading factor. This enzyme is also found in testes, seminal fluid, urine, plasma, synovial fluid and many other tissues. Hyaluronidase present in seminal fluid is responsible to facilitate fertilization by an outer covering of the ovum to permit easy entry of spermatozoa. Clinically, this enzyme is used to increase the absorption of fluid administered subcutaneously. It is also used as a local applicant to resolve post-traumatic swelling or edema. 2-chondroitin sulfates, it is the most abundant glycosaminoglycan in the body. It is found in the ground substance of the tissues, especially in cartilage, tendons, ligaments, aorta, etc. In cartilage, it binds with collagen to form a strong network of fibers. It can also form proteoglycan aggregates. The disaccharide repeating unit found in it is composed of N-acetylgalactosamine and D-glucuronic acid. The N-acetylgalactosamine is sulfated on C4 or C6. Figure 3.29 Structure of chondroitin 4-sulfate, glucuronic acid plus N-acetylgalactosamine. 3-keratin sulfate I and II. The disaccharide repeating unit found in keratin sulfate is composed of N-acetylglucosamine and galactose. It is the most heterogeneous group of glycosaminoglycans. It has a variable amount of sulfate present on C6 of either sugar. Keratin sulfate I occur in the cornea and is responsible for corneal transparency. It is also found in cartilage. It is linked to the protein via asparagine residue attached to N-acetylglucosamine by an N-glycosidic bond. Keratin sulfate II is found in loose connective tissues. It is linked to the protein by O groups found on serine and threonine residues. Figure 3.30 Structure of keratin sulfate, galactose plus N-acetylglucosamine. 4-dermatan sulfate, the disaccharide repeating unit found in dermatan sulfate is composed of N-acetylgalactosamine and L-geronic acid with variable amounts of glucuronic acid. It is present in skin, blood vessels, and heart valves. Figure 3.31 Structure of dermatan sulfate, E-geronic acid plus N-acetylgalactosamine. V. heparin, its repeating disaccharide unit is composed of D-glucosamine and D-glucuronic acid or D-geronic acid. The amino group at C2 and O group at C6 of D-glucosamine are sulfated. Acetyl group may also be found at its C2. The C2 of D-glucuronic acid is also sulfated. It is the type of glycosaminoglycan that possesses the highest negative charge due to the presence of a large number of sulfate groups. It has an average of 2.5 sulfate groups per disaccharide unit. It acts as an anticoagulant due to its antithrombin activity, by causing a release of lipoprotein lipase from the capillary wall. It is found intracellularly in mast cells lining the arteries of the liver, first isolated from the liver, lungs, spleen, thymus, skin, and blood. Figure 3.32 Structure of heparin and heparin sulfate, glucosamine plus glucuronic acid or E-geronic acid. 6-heparin sulfate. The structure of heparin sulfate is the same as heparin except some of its glucosamines are acetylated and it has few sulfate groups. Heparin sulfates are components of the plasma membrane of cells. They may act as receptors and may also participate in the mediation of cell growth and cell-cell communication. They are also found in the basement membrane of the kidney, and they play a major role in determining the charge selectiveness of glomerular filtration. Glycosaminoglycans are synthesized in the endoplasmic reticulum and Golgi apparatus. The polysaccharide chains are elongated by sequential addition of alternating acidic and amino sugars, donated by their UDP derivatives. The last step in the synthesis is the sulfation of some of the amino sugars. The source of the sulfate is 3-phosphoadenosyl 5-phosphosulfate. Glycosaminoglycans are degraded by lysosomal hydrolysis. They are first broken down into oligosaccharides, which are then degraded sequentially from the non-reducing end of each chain. Muco Polysaccharidoses are hereditary disorders that are clinically progressive. They are characterized by the accumulation of GAGs in various tissues such as the cornea, nervous tissues, spleen, liver, joints, heart valves, and coronary arteries. It results in various symptoms like skeletal deformities, defects in the extracellular matrix and mental retardation. Mucopolysaccharidoses are caused by a deficiency of one of the lysosomal hydrolyses normally involved in the degradation of glycosaminoglycans. This results in the presence of oligosaccharides in urine. These fragments can be used to diagnose specific mucopolysaccharidoses. Diagnosis is confirmed by measuring the cellular level of lysosomal hydrolyses. Some of the important mucopolysaccharidoses are summarized in Table 3.6. Chitin. Chitin is the second most abundant biopolymer on Earth, after cellulose. Chitin is a long-chain polymer of repeating units of the disaccharide acetylglucosamine. Chitin is composed of SS-1,4 linked units of the amino sugar N-acetylglucosamine. It is the main component of the cell walls of fungi, mushrooms and the exoskeleton of some major arthropods and insects. The chemical formula of chitin is C8H13O5N, N. Table 3.6 Features of important mucopolysaccaridosis. Mucopolysaccharidoses. MPS. MPS subtypes. Age at diagnosis. Mental development. Treatment options. MPSI. Hurler, MPSIH severe. 6 to 24 months. Impaired. After about 3 to 4 months. SCT, 2 years, ERT. Symptomatic. Hurler-Sky, MPSIH/S less severe. 3 to 8 years. Normal. ERT, symptomatic treatment. Sky, MPS is least severe. Greater than 5 years. Normal. ERT, symptomatic treatment. MPS 2. Hunter, severe, MPSIIA. 18 months, 4 years, depending on severity. Impaired. After about 2 to 3 months. ERT, SCT, symptomatic treatment. Hunter, less severe, MPSIIB. Normal. Symptomatic treatment. ERT. MPS 3. Sanfilippo A, MPS 3A. 1 to 2 years for severe forms. For milder forms, not until adulthood. Perhaps even greater than 50 years. Impaired after about 2 years. Symptomatic treatment. ERT in development. Sanfilippo B, MPSIIB. Symptomatic treatment. Sanfilippo C, MPSIIC. Symptomatic treatment. ERT in development. Sanfilippo D, MPSIID. Symptomatic treatment. MPS 4. Morchio A, MPS EVA. 1 to 3 years. Normal. Symptomatic. Treatment, ERT. Morchio B, MPS IVB. Symptomatic treatment. MPS 6. Marodiazlami, MPS 6. 2 to 3 years. Normal. ERT, SCT, symptomatic treatment. MPS 7. SLI. 0 to 7 years, depending on the severity. Impaired early. Symptomatic treatment, SCT. MPS 9. Hyaluronidase deficiency. Not known. Not known. Symptomatic treatment. Note, SCT, blood stem cell transplant, ERT, enzyme replacement therapy. 3.10.2.2 Glycoconjugates. These are the type of heteropolysaccharides that include proteoglycans, glycoproteins, and glycolipids. Proteoglycans. Proteoglycans are complex carbohydrates having glycosaminoglycans covalently linked to core proteins. They vary in tissue distribution, nature of the core protein, attached glycosaminoglycans, and functions. Their molecular weight range, 105 to 107. The proteoglycan monomers associate with a molecule of hyaluronic acid to form proteoglycan aggregates. The association is not covalent but occurs primarily through ionic interactions between core protein the hyaluronic acid. The association is stabilized by additional small proteins called link proteins. Figure 3.33 Structure of proteoglycan, source, tosgoncosmetics.com. This linkage is most commonly through a trihexoside, galactose xylose, and a serine residue, respectively. An O-glycosidic bond is formed between the xylose and the hydroxyl group of the serine residue. Figure 3.34 Attachment of glycosaminoglycan to protein. Sugars are linked to a serine or threonine residue of the core protein. A and B represent the sugars of the repeating disaccharide. All of the GAGs, except hyaluronic acid, are found covalently attached to proteins, forming proteoglycan monomers. These GAG chains are composed of more than 100 monosaccharide units and there are about 200 GAG chains per monomer of proteoglycan. These GAG chains extend out from the core protein and remain separated from each other because of charge repulsion. The resulting structure resembles a bottle brush. Figure 3.35 Structure of cartilage proteoglycan monomer, source, Lippincot's Illustrated Review Biochemistry 7th edition. Mammalian cells can produce approximately 40 types of proteoglycans. These molecules act as tissue organizers and they influence various cellular activities, such as growth factor, adhesion, and activation. Many proteoglycans have been characterized and named based on their structure and functional location. E.g., syndecan is an integral membrane proteoglycan, varican and agrican are the predominant extracellular proteoglycans while neurocan and cerebracan are found primarily in the nervous tissues. Other examples of proteoglycans include D-chorin, biglycan, beta-glycan, surglycan, perlecan, fibromodulin, etc. 2. Glycoproteins. Glycoproteins are carbohydrate protein conjugates in which the glycans are smaller, branched, and more structurally diverse than the huge glycosaminoglycans present in proteoglycans. The carbohydrate chain is relatively short, usually 2 to 10 sugar residues in length, although they can be longer, whereas it can be very long in the GAGs. In addition, whereas GAGs have diglucosyl repeat units, the carbohydrates of glycoprotein do not have serial repeats. The glycoprotein's carbohydrate chains are often branched instead of linear and may or may not be negatively charged. About half of all proteins of mammals are glycosylated. Many of these are plasma membrane proteins and secretory proteins. Glycoproteins contain highly variable amounts of carbohydrates. Example: the immunoglobulin IgG contains 4% of its mass as carbohydrates, whereas human gastric glycoprotein, mucin, contains 80% carbohydrates. 2 broad classes of glycoproteins occur in nature, the O-linked and the N-linked glycoproteins, figure 3.36. A glycoprotein may contain only one type of glycosidic linkage, N or O-linked, or may have both O and N-linked oligosaccharides within the same molecule. Figure 3.36 O-linked and N-linked oligosaccharides in glycoprotein. In O-linked glycoproteins, the carbohydrate is attached to its anomeric carbon through a glycosidic linkage to the O group of a serine or threonine residue. The O-linked oligosaccharides may have one or more of a wide variety of sugars arranged in either a linear or branched pattern. Many O-linked oligosaccharides are found as membrane glycoprotein components or in extracellular glycoprotein. The O-linked oligosaccharides help to provide the ABO blood group determinants. The N-linked oligosaccharides fall into two broad classes: complex oligosaccharides and high-mannose oligosaccharides. Both contain the same core pentasaccharide, but the complex oligosaccharides contain a diverse group of additional sugars, example N-acetylglucosamine, L-fucose, N-acetylneurominic acid, whereas the high-mannose oligosaccharides contain primarily mannose. Figure 3.37 Complex and high-mannose oligosaccharides, NANA, N-acetylneurominic acid, GAL, galactose, GLCNAC, N-acetylglucosamine, MAN, mannose, fucose, ASN, asparagine. Source: Lipinkit's Illustrated Review Biochemistry 7th edition. The biological advantages of protein glycosylation include improving the solubility of proteins, providing address labels directing the targeting in cells and between tissues, and protein folding and stabilization. Membrane-bound glycoproteins participate in a broad range of cellular phenomena, including cell surface recognition by other cells, hormones, viruses, cell surface antigenicity, such as the blood group antigens, and as components of the extracellular matrix and of the mucins of the gastrointestinal and urogenital tracts, where they act as protective biologic lubricants. In addition, almost all of the globular proteins present in human plasma are glycoproteins. Certain hormones like FSH, LH, TSH, HCG, etc., are glycoproteins. Other examples include immunoglobulins, antibodies, lactalbumin, pancreatic RNase, transferrin, ceruloplasmin, etc. 3. Glycolipids and lipopolysaccharides. In glycolipids and lipopolysaccharides, complex oligosaccharide chains are attached to membrane-anchored lipid moieties. The complex structure of the lipopolysaccharide found in the outer membrane of Salmonella typhimurium is shown in Fig. 3.38. These molecules, which are found in other gram-negative bacteria such as E. coli, are recognized by the immune system of vertebrates and therefore are important determinants of the serotypes of bacterial strains. Serotypes are strains that are distinguished based on antigenic properties. The fatty acid-containing lipid A portion of lipopolysaccharide is called endotoxin. Its toxicity to humans is responsible for the dangerously lowered blood pressure that occurs in toxic shock syndrome resulting from gram-negative bacterial infections. Figure 3.38 Structure of lipopolysaccharides, GLCNAC, N-acetylglucosamine, MAN, mannose, GLC, glucose, GAL, galactose, A or ABOAC, O-acetylabicose, R or RHA, rhamnose, K or K2, 2-keto-3-deoxyoctonate, H or Hep, heptose. Plants and animals contain many types of glycolipids and glycosphingolipids. The gangliosides are glycosphingolipids containing complex oligosaccharide chains with sialic acid residues. These lipids are important for cell-cell recognition, and some serve as receptors for the entry of bacterial toxins such as cholera toxins into mammalian cells. Other glycosphingolipids, the globosides, contain oligosaccharide chains that serve as the blood group antigens. The detail of glycolipids is given in the chapter on the chemistry of lipids. 3.10.2.3 Mucilage. Mucilage is a group of complex thick and sticky substances that are made by almost all plants and also by several microorganisms. Mucilage is a polar glycoprotein as well as an exopolysaccharide. In plants, mucilage has an important role in storing water as well as food, germination of seeds and also strengthening the membranes. Flax seeds as well as cacti, including other succulents, are exceptional mucilage sources. Important types of mucilage include agar, pectin, and vegetable gums. Agar, it is a type of mucilage. Chemically it is a sulfuric acid ester of a complex galactose polysaccharide. It is a gelatinous material derived from the seaweeds, specifically used as a culture medium for the growth of bacteria, as it is not liquefied by bacteria and other cells for diagnostic or laboratory experimental purposes. Agar remains undigested and has a stimulatory effect on the gastrointestinal tract. Clinically used as a laxative for the treatment of constipation. 2 pectins, it is a white, amorphous, colloidal heteropolysaccharide of high molecular mass occurring in the pulp of ripe fruits, especially in apples, carrots, and many citrus fruits. On hydrolysis, it yields galacturonic acid, arabinose, galactose, acetic acid, and 10 to 12% methyl alcohol. When heated together with sugar, it causes a thickening that is characteristic of jams and jellies. 3 vegetable gums, this type of heteropolysaccharide is derived from vegetables and plant sources. These are the complexes of hexoses or pentoses or both in glycosidic linkage with carbohydrate acid groups. An example of vegetable gum, the gum arabic, is composed of galactose, arabinose, rhamnose and glucuronic acid. They are used in the preparation of various drugs, confectionery, and also used as an adhesive. Summary. Carbohydrates are polyhydroxyaldehydes or polyhydroxyketones or the compounds which give them upon hydrolysis. They are the major source of energy in most organisms. They have a wide range of functions. Their general formula is CH2O, N. Their caloric value is 4.1 kcal/g. Carbohydrates are classified into 5 major groups: monosaccharides, disaccharides, oligosaccharides, polysaccharides, and derived carbohydrates. Monosaccharides are the simplest carbohydrates. They cannot be hydrolyzed further. Disaccharides are formed by the condensation of 2 monosaccharide molecules. In disaccharides, the monosaccharides are linked together by glycosidic linkage. Oligosaccharides are formed by the union of 3 to 10 units of monosaccharides. Polysaccharides are formed by the condensation of a large number of monosaccharide molecules. Homopolysaccharides are polysaccharides made up of the same kind of monosaccharide units, example starch, glycogen, cellulose, and dextrins, polymers of glucose. Heteropolysaccharides are polysaccharides made up of different kinds of monosaccharide units. Derived carbohydrates are obtained from other carbohydrates as a result of various chemical reactions upon them. Reducing sugars are those which have potentially active aldehyde or ketone groups in their structure. In other words, these sugars have free anomeric carbon atoms. The asymmetric, chiral, carbon atom is a carbon atom to which 4 different atoms/groups of atoms are attached, or all the valences are filled by different groups. Isomers are compounds having the same chemical formula but differ in their structural formula. For example, glucose, fructose, mannose, and galactose have the same chemical formula of C6H12O6 but their structural formula is different from one another. Enantiomers are a pair of stereoisomers that are mirror images of each other around the asymmetric carbon atom. Anomers, these isomers differ from one another in configuration around an anomeric carbon atom, which is carbon number 1 in aldoses and carbon number 2 in ketoses. Epimers, these isomers differ in configuration around one specific carbon atom. For example, glucose and galactose differ from each other only in the position of the O group around carbon number 4, so they are called C4 epimers. Optical isomerism is the ability of biomolecules to rotate the plane of polarized light either on the left or right-hand side. When a freshly prepared solution of D-glucose is observed in a polarimeter, it is seen that its specific rotation initially keeps on changing and then becomes fixed after some time. This phenomenon is called mutarotation. All those simple sugars, monosaccharides, which contain 6 carbon atoms per molecule are called hexoses. Glucose is an aldohexose, also known as dextrose or blood sugar. Glucose is a primary source of energy for brain cells and erythrocytes. Normal fasting glucose level is 70 to 110 mg/dl or mg% and random blood glucose level is 80-160 mg/dl or mg%. Glucose can be converted into a non-carbohydrate substance, example fat, and in this way regulate overall metabolism. Glucose may be synthesized from non-carbohydrate residue within the body via gluconeogenesis. Fructose is a ketohexose or levulose commonly known as fruit sugar because of its high concentration in fruits. Galactose is an aldohexose. Galactose is a type of sugar that is less sweet than glucose. It is considered a nutritive sweetener because it has less food energy. Ribose and deoxyribose are the pentoses, found in RNA and DNA respectively. Glycosides are the compounds having monosaccharides linked by acetal linkage of the glycosidic bond. Maltose is a disaccharide of 2 glucose units, present in malt and malt products. Lactose is a disaccharide known as milk sugar formed by 1 unit of glucose and 1 unit of galactose joined with alpha, 1 rightward arrow 4, linkage. Sucrose is a non-reducing sugar as there is no free aldehyde or ketone group, as the glycosidic linkage is found between carbon number 1 of glucose and carbon number 2 of fructose. Starch and glycogen are homopolysaccharides in plants and animals respectively. Both are polymers of glucose. Muscle glycogen is regarded as a fuel reserve for the synthesis of ATP during muscle contraction. Glycosaminoglycan molecule is an unbranched polysaccharide made up of repeating units of disaccharides. Proteoglycans are complex carbohydrates having glycosaminoglycans covalently linked to core proteins. Glycoproteins are carbohydrate-protein conjugates in which the glycans are smaller, branched, and more structurally diverse than the huge glycosaminoglycans present in proteoglycans. Glycolipids and lipopolysaccharides are complex carbohydrates in which oligosaccharide chains are attached to membrane-anchored lipid moieties. References. Exercise questions. MCQ 4. Which of the following reasons render the body to store carbohydrates as glycogen over simple glucose? It can provide many other monosaccharides. It doesn't disturb osmotic pressure. It has a high energy level. It is a high molecular mass polymer of glucose. MCQ 5. Which of the following monosaccharides is the catabolic product of sucrose? Glucose and fructose. Glucose and galactose. Glucose and glucose. Glucose and mannose. MCQ 6. Which of the following compounds is the heteropolymer of D-glucuronic acid and N-acetyl D-glucosamine? Chondroitin sulfate. Glycolipid. Glycoprotein. Hyaluronic acid. Proteoglycan. Please write answers in the comments. End of Part 2 of Chapter 3. Thank you.